Northeast Science Movement for giving me the privilege of sharing some of my research experience. Northeast India abounds in myriads of wetlands and they support specially adapted flora and fauna. Of the various faunal components, I have selected to speak on amphibia, as they are known as ecological indicator species. Amphibian survival depends on the quality of our wetlands. Wetlands are amongst the most productive ecosystems on the earth. They provide contribution to economy, livelihood to rural communities, offer a range of hydrological and ecological benefits, recharge and discharge of groundwater, flood control and sediment retention, clean water supply throughout the year, support a high level of biodiversity with specially adapted flora and fauna. Wetlands are ecologically sensitive zone and they are adaptive. Due to the water filtration processes which occur at wetlands, they are sometimes referred to as kidneys of a catchment area. Wetlands cover 6% of the world's land surface and contain about 12% of the global carbon pool, playing an important role in the global carbon cycle. In India, wetlands account for 4.7% of the total geographical area of our country. And in Assam, the total wetlands account for 2.2 lakh hectares, where floodplain wetlands are 1.12 lakh hectares. In India, we have two mega diversity hotspots. One is Eastern Himalayan region and the other is Western Ghats. They have certain characteristics. The Eastern Himalayan region has ultra-varied topography, species diversity and endemism. Concentration of species per unit area is more. Altitudinal variation is very prominent as well as climatic variation, and they are of zoogeographic importance. So this I'll discuss slightly more in detail. This area is situated at the confluence of Indo-Malayan, Indo-Chinese and Indian biogeographical region. So this map explains it. This part is the Eastern Himalayan region and this is the Western Ghat. High level of endemism in the Western Ghat is because there is no contiguous landmass. There is a barrier of this water. So the species here, they have evolved on their own since ages. And here we can refer to Gondwana theory. So here the Eastern Himalayan region is contiguous on one end with the China, it is contiguous with the Myanmar, and it is contiguous with the Indian subcontinent. The species, they don't know the political barrier. So here in this region we find Indo-Chinese fauna. In this region along Manipur, Nagaland and Mizoram we find the Indo-Burmese fauna or the Indo-Myanmar fauna, and here we find the Brahmaputra Valley region and the nearby areas, the fauna of Indian subcontinent. So amphibians, why they are important? Amphi means both or two, and bios means life. These amphibians, they have two phases in their life history. First is the aquatic phase, where the embryonic and the other larval stages are completed. It's completely different from the adult because during that time they undergo complete physiological and structural changes. So that phase is called metamorphosis, and the adult life cycle is just like any terrestrial vertebrate, and that is the reason this class of invertebrates are called as amphibia. They are important part of food chain both as predator, prey and biological control of pest. They are bioindicators of global climatic change and good barometers of environmental change that may go undetected by humans. When we say a species has become extinct, the process started 20 years hence, which we cannot know. Only the ecosystem experiences that change. And frogs are always being used in food and medicine. The amphibian fauna may provide an important indicator of the impact of anthropogenic disturbance to wetland ecosystems. They are characterized by moist, permeable skin, and that is the reason any contamination is in water. So these organisms are affected. They are cold-blooded. That means they are different from the warm-blooded or homeothermic organisms. They are called as poikilotherms. Because they are cold-blooded, during winter, they cannot tolerate the external change in temperature and they hibernate. This hibernation is an important phenomenon in case of this herpetofauna, both amphibians and reptiles. So amphibians lay cell-less eggs in water, which later hatch into gilled aquatic larvae. They are known as tadpoles. Aquatic larvae metamorphose into lunged adults that may or may not leave the water depending on the species. Worldwide amphibian declines were recorded by USGS, United States Geological Survey, since 2000. By that time, already in many parts of the world, amphibians started declining. There are certain possible causes identified on the basis of the studies conducted since 2000. One is pollution, then acid rain, pesticides, fertilizers, habitat destruction, introduction of exotic predators, greenhouse effect, ozone hole, effect of UV radiation on eggs, exotic diseases, and pathogens. One of these pathogens is Batrachochytrium dendrobatidis or chytrid fungus. Because of these chytrid fungus infection, there were declines in Australia, Central America, Europe, North America, because the chytrid infects the keratinized epithelium or the skin of the skin, and therefore the amphibians, they suffer from the disease and they die. During that period, all over India, surveys were conducted to record the incidence of this chytrid fungus. But Indian amphibians were not affected by this fungus. There was no record till then. So decline and disappearance were observed worldwide since early 1980s. Australia, it was recorded in 1999, Eastern North America in 2000, Latin America in 2000, and Central America, they recorded 40% disappearance. Declining Amphibian Population Task Force, DEPTF, was constituted in 1990, and they promoted studies on systematics, geographic range, population information, habitat preference, measured trait, conservation measures, and red list assessment. So this red list assessment was the result of Global Amphibian Assessment 1. That was in Coimbatore in India in 2002, and this is the result. Global Amphibian Assessment 2 is ongoing, and by next year probably we'll get the final results. In India, Zoological Survey of India in 2019 published the document where 432 species of amphibians were recorded, out of which critically endangered 19 species, endangered 33 species, and 19% were data deficient. That is very alarming, and not assessed by IUCN 39%. Data deficient species means except the identification of the frog, no data is available, like in what type of ecosystem they survive, what food they take, what is the development or the normal developmental stages of this particular species. Nothing was known about these particular species. So they are in the data deficient group. So faunal elements of northeast region, they have certain characteristics, like we find of the species available in India or the northeast of India, we find ubiquitous species. They are available throughout the country. Indo-Chinese species all along the Chinese border. Indo-Malayan species all along the Malayan border. Restricted range species, like certain species are there which are till now recorded only in certain specified localities. And there are certain national endemic, that means they are found only in India, not in any other country. Then South Asia regional, and there is a clear-cut demarcation of altitudinal distribution pattern, and this is very, very evident in Arunach Pradesh. So this is one picture showing how in water the tadpoles are developing. The adults, they laid their eggs as the water is conducive for their development. So here the tadpoles are growing. So this black colored these are all early stages, early larval stages of a species. So why do we need to conserve the habitat? So already I discussed a few points, like they have got naked skin, so they directly come into contact with the contamination that is present in water. So amphibians are indicators of ecosystem deterioration. Amphibian fauna may provide an important indicator of the impact of anthropogenic disturbances to wetland ecosystem. For example, if there are waste materials accumulating in any water body, or there is very high organic load in that system, not only amphibian, so many other aquatic organisms will be removed from that system. They will die and they will not be able to continue their reproduction. Habitat acidity has been linked to reduced population size and shrinking geographical range of amphibians. Concentration of metals in lakes may also affect amphibians. These are the studies of the scientists that have been referred in the bracket. Copper, zinc, and iron are all toxic to amphibians. Presence of amphibian in any ecosystem indicate the health of the ecosystem, and therefore they are known as ecological indicator species. If the habitat is suitable for amphibian, it will support many other species in that water body. So study of amphibia, this slide shows specially study of my team, that is since 94 we started our work, and till 2020 we have published a number of papers. In Arunach Pradesh, when we started our work, only 22 species were recorded, and by 2020 we published 61 species with 2 new records for the country. In Nagaland, only 10 species were recorded, and we published 34 species comprising 1 new genus to country and 7 new species for the country. And in Manipur, 13 species, and we studied 25 species and with 10 new state record. And in Assam, in Sibsagar district, detailed survey was taken up and 25 species were recorded. Sibsagar district is in the Brahmaputra valley, so the wetlands, they have all similar characteristics. From there, we found 1 species which is new to science. That is, that time when we published, its name was Rhecophorus safrai. Now it is Sangexcalus safrai. And from Kokrajhar, 32 species were recorded, and their study included a study on a group of frogs which are burrowing in nature. So after surveying all these areas, what we found that there are 4 categories. One is open tropical habitat, breeding in temporary water bodies. Some amphibians, during rainy season, there are certain temporary pools. So there are some frog species, they prefer to lay their eggs in those temporary water bodies. And disturbed habitat, breeding in temporary water bodies or permanent water bodies. Disturbed habitat means there are certain frog species that we normally see during the rainy season in and around our habitat. These are to some extent, they can tolerate the pollution of anthropogenic source. So they breed in our neighboring water bodies. Then open tropical habitat, permanent water bodies like reservoirs, ponds, and other water bodies which are permanent in nature, lakes, etc. And in forest habitats, the water bodies are comparatively cleaner. Running water as well as permanent water bodies, that means lotic systems as well as lentic systems present inside the forest habitats, they are comparatively cleaner than the water bodies that are near human habitation. So what did we actually study? Earlier studies were nearly only on one fine-shaped frog species, tries to compare with the existing literature and in the museums world over, and then give a name to the species, that is the taxonomic study. So I thought that if we do not have the basic developmental data of the species, it is not possible to measure conservation planning. So our study included, one is taxonomic study, DNA fingerprinting study, life history data, ecobiological study, and also tadpole taxonomy. Why the question of tadpole taxonomy comes here? When you visit any water body where frog species lay their eggs, so you will find only tadpoles. So when you collect one tadpole, it may not be possible to identify the species, of which species this tadpole is. It's very difficult because tadpole taxonomy is very complicated. So we did the tadpole taxonomy for the species that we studied. So for example, this is a Bacopharynx, scanning electron microscopic picture of Bacopharynx of Arunach. So this frog species was discovered in 1986, and the tadpoles or the female of the species were never described. So now in later slides I'll speak details about it. In this particular slide only I want to show that Bacopharynx has certain structures. These structures are species specific. This will not be similar to another species. So when we describe the tadpole of any species, this is very, very important, and this study shows that they have certain characteristics with which we can also identify the adult of the species. So ecobiological studies were on rare, threatened, endangered, and data deficient species. We did the water chemistry, that is the habitat data we recorded. May not be all the parameters of water, but the most important ones like dissolved oxygen, pH, conductivity, the ion, all the ions present there, and then food and feeding behavior. What the frog eats. There may be 10 species in one habitat, but if they are feeding on the same algae or the same aquatic invertebrates, then what will happen? There will be competition. So we found the food of the species, it is variable. That means in one pond ecosystem, if there are 5 species growing together, that means they are not competing for food. They have difference in their food habit. So food and feeding study is very, very important for each of the species. Then comes the reproduction. That means to prepare the normal developmental table. What the normal developmental table means? Like when the eggs are laid, when we get the next stage and the next stage, and when the life cycle is completed. So we monitor the reproduction right from egg laying till the young frog emerges from the tadpole. So study of normal developmental tadpole in selected amphibian species. So far we have completed for 12 species. Adaptive modifications in torrential amphibians. So that is again another subject, like why some amphibians we find in slow moving water, why in some lotic system, why in some lentic systems, why in some waterfalls, near waterfalls also we find some species. All these species, they have special adaptive modifications to remain in those habitats. So a few torrential amphibians were also studied and their habitat characteristics were also studied. So these can be found in our publications. So this is one just example from Nagaland, where from in the habitats, more than 100 habitats we studied, and there we found that the categories that I showed earlier, that species living in open tropical habitat breeding in temporary water bodies, then disturbed habitats, then in open tropical habitat breeding in permanent water body, species living in disturbed habitats and breeding in permanent water bodies, and then the forest species, they are present in lotic system as well as lentic systems. Lotic systems means the running waters, and the lentic systems means the stagnant waters like ponds, lakes, etc. So here the dissolved oxygen you can see, like in the category 1 we find dissolved oxygen level is up to 7.5. In 2, 3 to 6.5, so up to 7, again 6.8, and for the category where the lotic system is there, that means running water, the dissolved oxygen is 11.2. And in Nagaland, later where I am showing again another species where we found that dissolved oxygen level is 14 mg per liter. It's instead of oxygen, this will be one mistake here, that is it should be DO, dissolved oxygen, that is 11.2. That means very high level of oxygen is required for the tadpoles to complete their life cycle. So this is one, this completed its life cycle in our laboratory from egg stage till completion of the metamorphosis. So here you can see the small tail stuff that is getting absorbed, and this is here the tail has been absorbed. This is that earlier it is called Rhacophorus maximus. Now the name has changed to Smaragdinus, Zangixella smaragdinus. So how in water, certain parameters we record in water itself. At the time when we collect the tadpole or some life history stages, then only we record it. It is so temperature, water temperature, air temperature, pH, all these things are recorded at the moment. Then we collect the fixed sample for analysis of dissolved oxygen later in the laboratory. And then we also collect water sample so that we can record the trace elements with the help of atomic absorption spectroscopy. That is done in the laboratory later. So these are the two amphibian habitats where you will find different life history stages of different species that are using this habitat for their egg laying. So conditions of torrential stream. So here I want to mention about the torrential streams, like in waterfall. High speed water current, food availability is sufficient amount, stones and crevices provide excellent breeding grounds for the organisms and also provide hiding places from predators. Shallow and crystal clear water with high intensity of sunlight, and water is highly oxygenated. There we find 8 to 15 mg per liter. So if we want, we cannot, if we can recreate the conditions in laboratory, only then these sort of tadpoles can be reared in the laboratory. Otherwise, what we have done, in the habitat where all the ecological conditions are conducive, we make enclosures in the habitat and then record all the life history parameters till they complete the metamorphosis. So these are some of the habitats. This is the river Vasistha near Guwahati. This is the wetland of the Sabitry estate where we found a new species. This is a torrential stream in Wokha district, Nagaland, where we found a new genus for the country. So this is in Arunachal Pradesh. These are the areas in Arunachal Pradesh. So now we come to some species accounts. Just for example, all the 12 species it will not be possible to show here, but this is one of the first species we completed the life history study. This is from Nagaland, and they breed in temporary water bodies as well as permanent water bodies. So this is the amplecting male and female of the species in the habitat, and this is the Hyla annectens. This is the only hylid species found in India. There is no other hylid species so far recorded from India. There are other 5 hylid species in China. So these are the eggs covered in the jelly coat. And these are some of the stages I showed, where you can see the important landmarks in development, like from 1 cell to 2 cell stage. So we record the timing here. So we record the timing from the single cell till this completion of the metamorphosis, and that is called normal developmental table. So this is the laid eggs. So we keep them in clean water with food so that these tadpoles, they can grow and show their stages, and we record. In 2001, this particular paper was published. So in Kokrazhar, I told that we studied the burrowing frog species. These are the 4 species. So Teprobenica, Globalusus, Corolla asamensis, these are microhylid species. So here Microhyla ornata, some of the stages. So this way we keep photograph of the landmark stages. So this is a 16 cell stage. This is the mid cleavage. This is the dorsal lip structure, neural plate, and then closure of neural fold. As the vertebrate, in case of most of the vertebrates, the development is in the body or in ovo, in case of birds. So this is amphibian stages, or amphibians is the only group where we can see the development with naked eye under the microscope. And always in laboratories, to show embryonic developmental stages, always frog embryos are used world over for laboratory studies. So this is a species is very commonly available in Northeast. So we described it for the first time in 2003 from Nagaland. So this is Humerana hibernalis hydrophyllix leptoglossa. So we compared the development minute by minute for both the species, if there is any difference. So we found the difference in the total time taken for completion of metamorphosis, being in the same habitat at the same seasonal, in the same season. So this Humerana hibernalis, like for this particular species, some of the developmental stages I am showing. So here the development is ongoing development within the jelly coat. So from the jelly coat, when they hatch out, then we find other stages, then they become a free living tadpole till completion of the metamorphosis. So this is a new, these are the 2 new records for India, Humerana hibernalis, that we reported for the first time in 2003 and completed the developmental study in 2010. And this one is that in Nagaland, I showed one torrential stream where the oxygen, dissolved oxygen level showed 14 to 15 mg per liter. There we found this genus, and this genus is Leptobrachyla lateralis. So the frog species with all taxonomic details, it was published in 2008. This is one example of certain habitats. This is commonly known as, we call it as bamboo node frog. So in bamboo, in Nagaland, there are bamboo bushes. So in the bushes, what happens? Some bamboos, accidentally or naturally, there are certain holes. In these holes, during rainy season, water accumulates. So when water accumulates, there they lay their eggs and the tadpoles there go. So one such bamboo we cut open. So inside this bamboo also we found certain small newly emerged frog, and here inside the bamboo we find the tadpoles as well as froglets. So this is the species. So these species we have published in November this year. So this is a species I told earlier, that Arunach. It was published in 1986. So it has certain folds or wing-like structures. So it was named. That time nothing was known except the male of the species. And since 86, the tadpoles and female of the species were not known. So in Nagaland, as I said, in enclosed water body, we studied the life history of this species, and we found the only male has these modifications. The female is just like any other any other ranid frog. And so, because we found the amplecting pair together, we could describe the female of the species for the first time, and also we could describe the tadpoles with all details. And this is the newly emerged froglet, but you can see the tail still there. Its tail is resorbing. This is called as tail stub stage. Tail stub stage. So this species, this paper was published in 2006. So this is the new species we recorded from the Saffrye tea estate. So Zangixellus saffrye. So this species is new to science. So this is the genus Leptobrachyla. So for the first time, the presence of this genus in India was reported. Leptobrachyla lateralis. This I have already shown. This is a species we recorded from Arunachal Pradesh. That in 2001-2002 we published this species from Arunachal Pradesh, and this is Hemerina hemeralis that also was recorded in 2003. I already mentioned about it. Some of the amphibians from Tirap district of Arunachal Pradesh. Here, along with all these other species, the Zangixellus smaragdinus, one of the life history stage, the newly emerged froglet I showed earlier, and here we can show the Ichthyophis sandenu, this another group of amphibian that is recorded, but it is very difficult to find because these are burrowing organisms and they remain in burrowed in sand, so only very rarely we can find them. So this was recorded from Tirap district. Thank you. This is the Smaragdinus, Zangixellus smaragdinus species. Earlier the name was Rhecophorus maximus. So this one completed the life cycle in the laboratory. So this is the newly emerged froglet, and so I have used this photograph to say a big thank you for listening to my talk. Thank you.